So our laboratory studies how different hormones um, regulate the um, production of glucose by the liver, and what we're interested in is a group of genetic switches that turn on genes that are involved in a process known as gluconeogenesis. So normally when we fast, gluconeogenesis is turned on and glucose output is increased. It allows the brain to function because the brain depends primarily on glucose and not free fatty acids for its as a metabolic source. The hormone glucagon is released from the pancreas during fasting, where it stimulates a genetic switch called CREB. Glucagon stimulates CREB activity by promoting its phosphorylation in response to the protein kinase pKa. The phosphorylation of CREB increases its activity by promoting an association with a coactivator called CBP. CBP is an enzyme actually that acetylates histones on the promoter. And by acetylating histones, it allows CREB to come in and bind to the promoters that are involved in gluconeogenesis. In parallel with that pathway, there is a second pathway that's activated by cyclic AMP. This involves a group of coactivators called CRTCs. These are proteins that are actually sequestered in the cytoplasm under basal conditions, and they are activated by dephosphorylation. And so when they get dephosphorylated, they migrate into the nucleus where they bind to CREB in addition to CBP. So there's two pathways that are involved in the activation of the gluconeogenic program. The reason this is interesting is that this pathway is super activated in insulin resistance, where it contributes to the overproduction of glucose by the liver. So we think this is going to be an important area to focus on for drug development, because by lowering glucose production, we could avoid a lot of the sequelae that are involved in type 2 diabetes.